want to welcome you to this webinar and thank you so much for joining. As you are connecting, please find the chat. It's an icon at the bottom of the webinar. So find this chat icon because I'm going to be asking you some questions and interacting with you through the chat. So go ahead there and drop us a hello and let me know how you found out about this webinar. Was it social media? Was it word of mouth? Do we work together? I don't know, where else? How did you find out about it? And today we will talk about personalized medicine for rare disease, Robin's disease. And you probably already know who Robin is. It is Typhaine's son. Before we go there, a few words about us. So chances are, as I said, that you know at least one of us, but for those who don't, few words of introduction. I'm Alexandra Zhurov, and I'm the host of this webinar, and Typhaine Lejeune is going to present on today's topic. She is the expert, and she's Robin's mom. And I will ask her to give a few words about her background and her scientific expertise in a minute. We are both board-certified pathologists. We do toxicologic pathology. This is our specialty, which is supporting drug development. We both work at Charles River Laboratories, which is a contract research organization performing preclinical studies supporting drug development. And this is also how we know each other. We are colleagues and we are also friends. And here, a short disclaimer, we both work for Charles River and we're going to be talking about things from Charles River, inspiration, and a lot of Charles River is going to be in this webinar, but this is not a Charles River webinar. This is our private webinar where we want to share the message and the approach for Typhaine's message, Typhaine's story, which is a powerful one, and a lot of science, pretty cool science that I was not aware of before I learned about it from Typhaine. So, okay, we are colleagues, but why is it the two of us and not Typhaine and another colleague or Typhaine and someone else? It's Typhaine's story and she's going to be talking. Why am I there even, right? So, so first of all, I have the blog and I have a platform to host such a webinar, which I thought would be great if I could offer to Typhaine. But let's take a couple steps back and where we met, how we met. We met at Charles River in Montreal when I started the job there. And if you watched a few of those videos on LinkedIn or on social media that I was posting to let you know about this webinar, you know that when I joined, I was 8 months pregnant with my first son. So my colleagues would give me gifts for baby gifts, including clothes. And I remember this one dark blue snowsuit that was from Typhaine. I think I got it from Genevieve, but she got it from Typhaine. So this is like a hand-me-down through multiple colleagues. No, I got. Anyway, hand-me-down from multiple colleagues, and it made its way to my son as well. This was a winter baby, and it snows in Montreal from November to March. I think, Typhaine, you told me yesterday there was snow as well. So when my son was little, he had constant ear infections and he ended up having tubes. So this is a surgery. So when I was kind of stressed. It was beginning of my job there. So I would talk to colleagues and said, oh, I would, and to Typhaine as well. And it turns out that we used the same pediatrician. And then later my son ended up in the same hospital as Typhaine's son. So Typhaine is a very private person. And if not for this connection that we had with the kids and with health issues, I would probably not know about the fact that she's raising a son with disability up to the fundraiser that she started. She now has a fundraiser where there is a longer description of her story. So before that, that was not public knowledge. So when I noticed that she went public with it and that there is an option to do something, I wanted to help. Long story short, our parenting stories crossed a couple of times. So when she shared it, and not only did I want to donate, which I did, but I wanted to spread the word, because one person can only donate so much. Many people can donate a lot more. And also, I was super curious how she is doing this. There were a couple of stories that Typhaine is going to mention in our company at Charles River that had a similar background. So without further ado, I think this is enough of introduction. Typhaine, the floor is yours. I mute myself and I will be very respectful when I will want to interject. I will raise my hand, virtually raise my hand. So go ahead, Typhaine, and tell about yourself first, because I left this for you.
Thank you, Alex. Well, I think you said quite a lot already, actually, because obviously we have the same scientific background. I have been working for Charles River now for 15 years, and I am a pathologist over there. So this leads me to actually a small disclaimer also myself, which is that what I am sharing today is what I know of the science that is available, and it is my view of it. I am not saying that everything will be accurate. I hope it is, but this is really what I have learned so far. I have the scientific background, yes, but precision medicine is not my field. So I am learning. I am trying to learn as fast as I can, but I am learning. So this is what I'm sharing today. So let's go.
Okay.
So, yeah.
Let's start with the first year. So we framed it, you are raising a child with disability. How was it the first year? Tell us about this. Like, how did you tell us about it?
Well, I guess the short answer could be nice and awful. Because I mean, it's the first year of a baby, right? So it's nice. I mean, the first years of a baby are always nice, but obviously it was not exactly the same for us. So Robin was born in July 2014, and my pregnancy, it was my second child. His brother is just 2.5 years older than he is, and you know, pregnancy was just fine. Everything was fine. He was late, just like his brother, 1 week late. And birth went fine as well. And well, you know, at birth, they take scores, of how the baby is doing. And he was doing, he was on the low limits. So, but still he was fine. The only thing was that he had to be kept under oxygen for 24 hours because his oxygen levels, blood levels would just like go down anytime he would cry or eat, so pretty much anytime he would need to do anything else but breathing, his oxygen levels were going down. So he stayed for 24 hours like that. The pediatrician found that he had some hypotonia, so low muscle tone. But then he thought it was getting better, so after 3 days, he was released from the hospital and we went back home. But then his first, we get the first visits, right? 1 month for kids, it's 1 month, 2 months, 4 months, 6 months. So 1 month, everything was okay according to the pediatrician. 2 months, we were to another pediatrician because our whole family is back in France. So that's where we were at that time. And that pediatrician said he had a torticollis, and he was quite also on the, had low muscle tone. So we did some physio over there. I mean, the pictures I put on there are actually from his first 6 months. And actually on this one and on this one, the torticollis was fully installed, and you can see that he's not very big, but his head turned upwards like this towards the right side. That was the torticollis, and he would pretty much keep his head like that most of the time. And so after 4 months, 4 months, 6 months, same thing. I mentioned to the pediatrician there was a torticollis, but he was not acknowledging it particularly. But thankfully, 3 days after our 6 months visit, we were going to the physiotherapy department of the hospital, and I will just tell you afterwards how we came there, but he was diagnosed there with like a severe torticollis with plagiocephaly, which is a flat head, and also a severe hypotonia. So that's when all the referrals started, actually. So first we needed to get a helmet for him because the head was so flat behind. It was on his right side. So this picture actually shows that when you get a flat head on one side, actually at some point it gets so severe that the face itself is also deformed. Like the right side of his face was pretty much going forward. If I would look at him from above, I could see that his ears were misaligned, but totally misaligned. Like the right ear was much forward than the left ear. And so you need a helmet for that, because the helmet will just put pressure on this part of his face, release the pressure here, put pressure here to really reshape the head. And so that's what he got at 7 months. And then 8 months, he had surgery because one of the things he had at birth was, yes, Alex.
So the moment where you went to the hospital and he was, the symptoms were noted by a physician, that was 6 months, or that was the 7 months?
It was 6 months.
Okay,
it was
so
6
then
months,
at 7 he already had.
Yes, and they advised me to actually get a helmet. So that's the time it just took to, you need to get a mold of his head to get the helmet really sized for him. And this helmet, he actually kept it for 3 months, and it's a 23 hours per day that, well, pretty much you take it off for bath. That's pretty much it. But thank God he seemed to be okay with it, so that was okay. And so what I was saying is that what he had when he was born was cryptorchidea, which is an undescended testis. So they want to usually have surgery for that kind of congenital thing as fast as possible, because if not, you just lose, well, you don't lose the testis itself, but you lose the ability to actually make spermatozoids in the testis if you don't have it descended. So he had surgery, and I can honestly say that to that day, we are really thankful for that congenital thing, because this was our door into the healthcare system. For those who don't know Quebec healthcare system, it's good, but it's very difficult to get in. So it was really our door in, because he was referred right away from birth to urology for this. And then in urology, they noticed that there were a few things that seemed to be a bit off, but they were not sure. Yes, Alex?
Very hard to get in, especially if you're from a different country.
And so we were referred to the general pediatry over there, and it's them who took us to the physiotherapy department. So that's why everything happened. And then from physiotherapy, all the referrals came in: occupational therapy, speech therapy, etc. So in a way, we are thankful for that. And the surgery went very well, and it was fine. So a few of his milestones, just to give you an idea of what developmental delay can lead to, because obviously it happens a lot that you get, you feel like your kid is a bit late maybe, but everybody around you tells you like, don't worry, it will come in time, but at some point you actually need to worry. So pretty much a baby usually holds its head around 3 months of age, and at 6 months he was not holding it properly. He was still tilting like some babies do, you know, when they're almost holding it but not quite. They usually can stay, you know, be put in the seated position and stay like that around 7 months, and he could do that at 11 months. And I like this photo actually, because you can see how he is spreading his legs. This is really to stabilize himself, and those are the things you learn when you do physiotherapy, is that at some point he had to wear special shorts that were, the thighs were sewn together to prevent him from doing this, and really make him use his trunk to strengthen his trunk to stabilize himself, rather than just spreading his legs and just stay like really like that. So those are the things that always amaze me with the different therapies, is that they have so many great ideas. So that was one thing. And then this one is, yeah, certainly an important one, because this was the very first day when he actually decided that he would start sort of crawling. I really like this video because we had been waiting for that for a very long time, and he is, you know, he's happy about it also. That's kind of cute. So he was 16 months old when usually this happens around 10 months old.
I love this video because his brother is cheering on this
exactly.
on this
He's
blue
attracted.
stool.
Yeah, he did it because he was attracted by his brother actually. So that was the reason. And so we started also at some point some rehabilitation centers, because you cannot do long-term physio or occupational therapy at the hospital. They only do short-term. So they referred us over there. And he started it was September 2017. So we got into some more milestones, thanks probably to the rehabilitation center, because they pretty much, it was a really new vision for us, because they are really focused on the parents, which was very nice. And they pretty much ask you what is important for you and for your child at that very moment, because obviously you need to choose your battles. I mean, he needed help in everything, gross motor skills, fine motor skills, speech, feeding. So at that time, you know, he was a bit more than a year old now, September. So he was 15 months old, and we really focused on physiotherapy because we were actually wondering if he would ever walk one day, which is, well, not something you really want to wonder about your child. So, and so this is so May 2016. So he was starting, you know, he could hold on to furnitures and, you know, do a few things. That you will see how he spreads his legs again to really stabilize himself, like right there. But finally, he really had his first steps, you know, around 2 years old, which, you know, is not the usual as well. And I put the feeding picture on the other side because feeding was certainly a real challenge. And I mean, we are lucky because some kids with Cat6b syndrome need a G-tube actually to be fed. A lot of them for at least a few years. Some of them keep it their whole life. So he finally got to feed by himself, but he would choke on milk when he was a baby. We started puree, you know, around 6 months. He would also choke on it, and really not a lot of it. But I mean, babies not liking some foods, I mean, that's not unusual. But we really didn't start solid food before he was 18 months old, because it was too, it seemed to be too dangerous for him. So then next one, sorry. So then we reached 2 years, as I was saying, and we got to start another program, because that's what you keep looking for. You keep looking for therapies, you keep looking for different programs that can be offered and that can improve, you know, because therapies are really the only way to go to improve their motor skills. So we found this one that was at our aquatic center, which was really a one-on-one program, and it was half gym, half swimming pool, which was really great for him. Those pictures are not from the beginning, because he was certainly not capable of doing this when he started it, which was September 2017. But this program has been great. And actually this program had been our Sunday afternoons until COVID happened, because again, it was a great program. You know, one-on-one programs are not easy to find, but they are the best ones for sure. And we quickly actually realized that he loved water. And that's one other thing, is that you need to find what motivates your child, because those are the best ways to really improve them, you know? So we definitely took advantage that over the years that he actually liked that water. And see, I put, you know, this live picture is now, you know, it was this year, because he's still very much into it. He doesn't know how to swim, but that's fine. You know, he keeps improving, and it's very good for muscle tone. I'm guessing he likes water because it takes less efforts from him, you know, to really move how he wants to move. Because that's the thing. I think a kid with low muscle tone, they will always go for the shortcuts. They will always go for what takes them the least energy, right? Which is logical. I guess we would all do this, right? That's what I was explaining about the sitting position. You know, he prefers spreading his legs rather than really using his trunk to really be steady. And that's the same for everything. He prefers to skip mastication if he can and just try and just swallow the food. But that's what also takes him, you know, to choking sometimes.
So before we talk about the diagnosis, I want to ask, so if you guys have, and gals have kids, write me the number of kids you have in the chat. And then before we move on, I remember I joined, you only were working 4 days a week. And I was like, that's so cool. Colleagues can work 4 days a week. And this was before I knew this, and I learned this, and I'm like, I'm glad I didn't tell her that it's so cool. I'm like, I was so glad I kept my mouth shut then. And then I quickly realized it's not fun, because the therapies take, well, you probably could have spent 4 days there instead of just 1 day, but that was the reason why you were not working. And then the other, and I only like mentioned this to you when you were working 5 days a week back, when I came back with my kid already, and then I learned this. I'm like, I was glad I didn't mention that before. And then another thing was, so I don't know, the other way, we were just chatting, preparing this webinar, and I was complaining about my kid throwing tantrums. And then Typhaine tells me, oh yeah, the terrible 2s and 3s and 4s. And actually, Robin only stopped doing this year. And I'm like, girl, stop complaining. This is a totally different universe. So basically, what you just said, right? You talk about that this is your reality. This is, I hope it's nobody else's reality here. It's not my reality. I still, you know, complain about my kids when they're throwing tantrums and don't want to do what I want them to do. But this, what Typhaine is talking about, is a total different universe. So just, you know, whenever you lose patience for your kids, think about this. So, okay, diagnosis. When and how did you learn what the disease was? You already mentioned the name, and we know it from the promotion of this webinar, but when was that you had the symptoms diagnosed at 6 months and you started doing stuff, but when did you know what was causing the symptoms?
Well, so back in 2015, among all the hospitals appointments that we were going to, one of them at some point was genetics, because developmental delay, global developmental delay usually points to a genetic cause. So if nothing else, was found. So we were there in December 2015, and I have to say that it's not easy. It's not an easy task for geneticists. I mean, he didn't have any particular features, physically. So they didn't really have anything to work on apart from global developmental delay, which is a very broad diagnostic and clinical feature that you can find in many, many disorders. So they did a bunch of testing and nothing, nothing came back positive. So pretty much they told us, come back in 1-2 years and, we'll see from there. Maybe there will be something new by then and we'll see from there. So we went back in June 2017. This is actually a picture from it. I liked it because, he has his Legos, like we were getting used to it. He was fine, the waiting room and everything. We had our habits there. So I thought it was a bit funny. And they did an additional testing, which is whole exome sequencing, which has not been available for that many years, this technology, which is pretty much not sequencing the whole genome, but sequencing all exons, which is the exons are the part of the genome that is actually expressed, all that is not expressed, but that serves other purposes, it's just left behind. So the whole exome sequencing, that's what it is. And that's what came back with mutation in the CAT6B gene, which was hypothesized to be probably pathogenic. And so they had to test both of us, my husband and I, to make sure we were not carrying that variant, because since this variant was only on one copy of his gene, one allele, that meant that the disease was dominant, right? And so if any of us had carried the variant, it meant that that was not the cause of it, because we would have it, one of us would have it. So we obviously did not carry that variant, so it confirmed that this is what it was, and his variant actually is also called Seybar-Burby-Secker, from, you guessed it, 3 people who diagnosed it. Well, back in the 80s, actually, but the origin obviously was not known at that time. They just diagnosed this broad syndrome that had developmental delay and intellectual deficiency and some congenital malformations. But I also just want to take 1 minute here about diagnosis, because this is a huge challenge for rare diseases, the diagnostic part, and it's difficult. It's very difficult nowadays. The average age for a child to be diagnosed is actually 7 years old. And so imagine the time that you lose versus being diagnosed at birth, from any disease. And this is certainly what, all patients, organizations, apart from working on research, they also work on diagnosis and having the genetic community in general aware as much as possible of, all these rare diseases so that they can be diagnosed as early as possible. So CAT6B syndrome, it has been divided clinically, although all clinicians do not necessarily agree on that, but in 2 syndromes, Seybar-Burby-Secker and genitopatellar syndrome, but they are really overlapping. This is what the colors mean on this chart. They try to really classify them either as Seybar-Burby-Secker or genitopatellar syndrome, or kind of an intermediate between both. And what you see here is a chart of the gene itself and all the genetic variants. So pretty much all the cases that have ever been published are fit on this chart. So they're all there. I circled Robin's one in red here, but since then there has been another publication recently this year that just found 10 more variants. but this is all the cases that exist. Some of them, now there are some clusters that appear, like maybe 4 or 5 patients having the same variant, which maybe points at some kind of mutation hotspots within the gene. Yeah.
So you're saying on this chart, there are all the cases that exist of this disease. So it's like, yeah. I don't know how many.
It's around 100.
Okay.
It's around 100. Okay. So, you know, you can obviously add to that cases that have not been reported, diagnosed, but not been reported. So, but we are probably not aiming at more than 200.
That is a very rare disease.
Yeah, we qualify it as ultra rare, pretty much. And so, well, to help geneticists, you know, diagnose, there are criteria that have been classified into major and minor criteria. So I won't really go through all of them, but pretty much if a child has 2 major criteria or 1 major and 2 minor, you should investigate and try to find if there is a CAT6B mutation. That's what it means, you know. It's not meant to be, oh, to confirm that your child has that. No, it's really meant for geneticists to, you know, point towards the syndrome. So of the major criteria, actually, Robin had this genital anomaly, which was his testis undescended, but then he has like a number of those minor criteria. So he definitely fits in.
Mm-hmm. So thanks for everyone who shared, for those who shared how many kids you have, and for those who don't. Thank you so much. One question to you, I guess it's personal, but I guess it very much fits into our webinar. Do you have, are you raising a child with disability, or do you have a child with disability in the family? Let me know in the chat if this is your universe as well. It's not mine, but it is Typhaine's. So
I think everybody, my story from really my family's point of view is common to everybody. I think we all have the same stories. Anytime there is a disability, you go through the same emotional steps, you go through the same overwhelming appointments and everything. So yeah, I
love the piece. I don't know if it's a little story or a poem that you have on your fundraiser about Holland, yes. Going to Holland. That I think it depicts very well the journey to somebody who is not on this journey.
Yeah, it's I love it very much as well, because it is such a great metaphor, and because you totally find yourself in a whole other universe that you had no idea existed. You know, I took him, before he went to school, I took him to this rehabilitation center that was for kids with motor disabilities. And I would see in the hallways, you know, I would take my baby at that time, and I would see in the hallways small kids in wheelchairs, with all sorts of, you know, disabilities. And that was quite something. That was really a slap in the face for me, because like stepping into this world is very particular. Now, obviously, I mean, we're used to it, but definitely that's just a parallel universe that, I mean, if it doesn't happen to you, you just have no clue that it exists.
Exactly. Totally. So what was the turning point? You said, oh, you had your routines when you would go to the waiting room. So you knew it was there to stay. You even knew what it was. What caused you to start having hope about the treatment? 100, 200 or up to 200 people with the disease. Like, how did you come up with this?
Yeah, well, you know, for sure, with my background and the genetic classes I took, you know, in vet school, to me it was just simple. I mean, it's a genetic disease. There's no treatment. It's genetic. It's his DNA. That's just who he is, and that's it. You know, the only thing we can do, and the best thing we can do, is just like support him. You know, take him to therapies and help him, to just improve as much and reach his, full potential. And in 2018, early 2018, what happened at work is that our sites actually worked on a study that was going to enable a little girl to get a new drug for her disease. And I think a lot of people, I think everybody in the pharmaceutical industry knows her. It's Mila. And she was going to get this drug, but it was the very particular thing about it is that this drug was designed for her. There was no other kid that could benefit from this drug. It was just for her. And because she had Batten's disease, which is a neurodegenerative disease, and she was 6 years old already. So at that time, so she had been showing, signs of degeneration, neurologic degeneration for a few years already. And her parents, partnered with the Boston Children's Hospital to design this drug for her. And I thought it was just amazing. And also the timeline of it was just amazing to me because it pretty much happened within a year. They identified her variant, they identified what was wrong with this variant, and then they, designed this drug for her. And at the end of the year, we were doing the toxicology studies, and then she got her first dose in February 2018. So it was, to me, it was really a crazy thing that I was just discovering, actually. And so I really started wondering what was possible, about it. And up till now, I had really focused on Robin, his therapies, and taking him there, like I was saying, supporting him, because I didn't think there was anything to be done on the scientific side of things. And I was putting all my energies in those therapies. But all of a sudden, this totally changed my view of things, and I realized I could put some energy in that. And I think with a child with a disability or rare disease, I think everybody has their own coping mechanism, and mine is really to be very active. I need to look for things, I need to search things. So until we had a diagnosis, I was just looking for therapies. I was looking for many ways to try and help him. And I actually had pretty much put a cross on the diagnosis because it had been 3 years. So I didn't even think we would have one, but then we got one. So I put some of my energy into looking into Cat6B, what it was and what other kids were like, but that's it. I didn't look more about it. Yeah.
I have noticed this like last week. I remember you were, we're going to talk about it. You were exploring the amount of money it would take, and it was one amount at some point, and then it like tripled or quadrupled the next. And there was one day where Typhaine was, oh, I have to wrap my head around it. And the next day in the morning, like, I have a plan. I explored this and that and check this and that. And yeah, we have to do it like this. So yeah, definitely very active approach to problem solving.
Yeah, well, that's again, I think we all have our own way to deal with it. And that's my way, that's what keeps me really going. And so, yeah, Mila and Milacen, because that's the name it was given, was really a new thing. And I thought I could put my energy into something like that, maybe, And so what it was, just briefly, it was an antisense oligonucleotides, which I think a lot of people are familiar with, but briefly, because I've always thought it was a brilliant idea. We have the DNA is transcribed into RNA, which is then translated into a protein. But then those oligonucleotides are small pieces of RNA that will just interact with the RNA within the cells. And so if you choose the right sequence, for this oligonucleotide, it will just bind to the RNA you are targeting. So the gene of interest in our case, right? And so it just acts, it will either pretty much from different mechanisms, it will end up in less protein production because it will just degrade that RNA. It would induce the degradation of that RNA that you don't want in a cell. There is also another mechanism in cells, which is RNA splicing, meaning the DNA is transcribed into RNA, but all the parts of this RNA are not useful for the translation into a protein. So some parts are just taken away, and that's the splicing mechanisms. And in some kids, like Mila, actually, her variant was a splicing defect. The splicing was not being done correctly. And the oligonucleotide they synthesized was directed at that splicing defect and was correcting it. So yeah, so that really, I really looked into those oligonucleotides, and I was really, really interested in them because, again, I thought that was a brilliant idea. Nowadays, I have come to the conclusion that it's not for Robin specifically, but I'll explain why later. And so back to our story, because, time was passing by, right? And he was growing. We started in 2017 this second rehabilitation center, which had this program for 3 mornings per week. We would take him there, and he was getting all the therapies there as well. So it was 2 places, which is why I was working 4 days a week, right? I would take him, Monday, Tuesday, Wednesday, taking there. At noon, taking him back to daycare for those 3 days. And then usually on the Friday, I had the other rehabilitation center to go to, and hospital appointments among that. So that's why I was just not technically working 5 days a week. So that's why I had to step down. It almost actually took me down at the end of 2017 because I was just not capable of doing it all. That was just way too much. At that time, my husband decided to stop working, which he did. For 2 years, he stopped working to take care of Robin because it was just too many things. And that is another thing that I want to point out, because that's the difficulty for families. Sometimes, a lot of the time, at least one parent has to stop working, and I'm not mentioning monoparental families. And that's difficult, for income, for a lot of reasons. So I bless my husband for that, certainly. And so this is just to explain the middle picture. At 5 years old, summer 2019, he graduated from that rehabilitation center, and this educator that is with him has been with him for 3 years, and she has done so many amazing things. Like she was the most amazing woman. I mean, I cried so much when I told her goodbye, at that time. It was insane. But yeah, so I just wanted to acknowledge her because it's been great. And so he's been growing up, so we were starting to be able to do some of the things that we used to like hiking. We like the outdoors. So, we went hiking, we went to Boston, which is the other picture, and he needs, obviously he needs to be held, or we still use the stroller or things like that. But overall, it was nice to be able to do those other things again. And progress, I just wanted to point out that progresses are really what keep us going. And I like those 2 movies because, you would never imagine, I would never be able to tell you when my first son was able to blow. Blow is a so difficult thing to achieve, and you would never guess it. And so this was his 4th birthday, and he needed to use a straw, but I think he was more surprised at our reaction than actually blowing his candle, because we were just like, yeah. So that was 4 years old, and that's his last birthday, So this is just what has been done, you know, in 3 years. So we are, you know, very proud of him for that.
So thank you everyone who shared things in the chat. I'm not gonna read them. They are pretty vulnerable. A couple of people shared, so thank you for that. And you have your own universe to live in. That is tough. So, Typhaine, what was the trigger and made what you said in vet school, you know, genetic disease is genetic disease. There is nothing you can do. Then I remember this Mila story. Let's call it the Mila story. Why I call it the Mila story? Because there was, obviously it was a great success and there was a lot of communication in the company about it. And I was like, what is this marketing video? Why do we like get this? I did not work on this drug. What is this? But then at some point, our manager sent this and said, oh, this is really worth watching. I'm like, okay, I'm gonna watch. And I watched and I cried for like 15 minutes as a mother of young little kid. And whoever watched it, probably they and have kids or wanted to have kids or, you know, care for little kids, probably cried a lot. So yeah, that was definitely a milestone for neurodegenerative disease. What was the trigger for you? What made you look more into Cat6b?
Well, another study.
We kind of have these at work. Good.
Yeah, and I guess we're lucky for that, you know, because we are in this field. And it was another antisense oligonucleotide, and this one, it was in 2019, and this one was designed for Angelman syndrome. And for this girl in particular, Quincy, it was not only her, you know, because this was not a unique variant, but this is the girl that we saw, you know, at work, Quincy and her mother, Allison. And I thought it was so inspiring because it was actually from the foundation. Angelman syndrome has a foundation. Two mother, Allison was one of them, decided to create a small biotech to support the development of this one oligonucleotide to go into clinical trial. And I was just like amazed that, you know, two mothers, they decided to create their own biotech just to support a compound. And I thought it was very, very inspirational. And what also struck me is that Angelman syndrome is not a neurodegenerative syndrome like Batten's disease or others. This one is a neurodevelopmental syndrome like Robin's, you know. It bears some similarities, you know, global developmental delay, speech delay, you know, a lot of things, intellectual deficiency. So I related even a bit more to that. And so I really started at that time looking into really basic mechanisms, like what this enzyme was doing, Cat6b. And so I had to dig back into my genetic classes again, because they are histone acetyltransferase. So histones, those are the yellow proteins that you see. You know, the DNA is this, you know, double helix that I guess everybody knows. But then DNA has to be compacted into chromosomes, and histones, that's what they do. They help, I'm sorry, I have the light. They help, you know, compacting the DNA, because the DNA wraps around the histones and, you know, to compact everything. But what has been discovered also at some point, and not that very far back in time, is that they're also important for regulations of genes. We used to think that histones acted essentially as DNA, like suitcases, you know, to hold the DNA. But it was actually clear at some point that histones, you know, they are regulators, and they have a lot to do with when genes are turned on or turned off, which is obviously a very important thing, right, for gene expression. So what I found a metaphor that I liked, which is like you can see them as a regulated suitcase that determines when the suitcase is opened and a gene gets out, right? And so they have a very important function, not only for the structure of the DNA. And so in particular, Cat6b stands for, it's part of a big family of histone acetyltransferase, like I was saying, the HATs. But then the K stands for lysine, the amino acid. So it is precisely a lysine acetyltransferase, meaning it adds acetyl to lysine residues that you see that are those tails here on the histones. It adds those groups, and this unwraps the DNA, and so allows the genes to be expressed at some point. And then we have their counterpart, which has deacetylases that then wrap again the DNA, and so genes are not accessible anymore. So it's always, there's a constant balance between those two enzymes. And there are many other enzymes, some will methylate, some will add butyryl groups. There are many enzymes like that modulate histones throughout the body. So it's a very complex thing. And so that really led me to really understand more. And in late 2020, I actually came to find on the internet that there existed a Cat6a Foundation. So I was curious about it, and I just sent them an email and asked them if they knew about Cat6b, if they knew about oligonucleotides, if that was something they were looking at. And they told me that they didn't, but they would be interested in me giving a presentation about oligonucleotides, which I did, and in January. And so after that, they actually asked me to join the board of directors of the foundation, because they were, Cat6a has 330 cases around the world. So pretty much they wanted to join forces, and create a slightly bigger community to be able to, eventually have more impact on research. So that's what I did, but it gave me access to a whole bunch of researchers that I didn't know about. And I got to speak with them a lot throughout the year. Some, generously shared their work that was not published yet. It really led me to believe that maybe something could be done,
So what was the process? How did you decide to start designing your own project?
Well, some accumulation. I wanted to really acknowledge those kids that we have seen. Mila, unfortunately, she benefited from the treatment, but unfortunately it was too late for her. So she lost her battle last year. But Quincy is doing better. She's on clinical trial. There is also Evie, who had a bone disease, and she was not supposed to live more than a few weeks after birth, and she is now 8 years old, And Michael is a very cute boy that is ongoing. His parents are also looking for a cure for him. So those, at some point, there was the last straw for me, which I just thought, okay, there must be something that can be done, and from the talks I had with researchers, there seemed some Cat6a patients, had been supplemented because there seemed to be hints of metabolic disease in them, mitochondrial disease. So, and those supplements had done some improvements, and another researcher had tested those supplements on Cat6b mice, and the mice had improved as well. So, I was just thinking supplements are such an easy thing, I at least want to try that, but at least determine, the concentrations and things like that, for it to be precise. And so that's really when I started engaging, CRL, Charles River folks, that work on rare diseases, and they put me in contact with other people outside of our company. And I was really doing those talks in the first place in the name of the foundation, because I was thinking maybe, the foundation has some research budget, a small one, because it's a small foundation, but they finance a couple of researchers in academia. But I was just thinking maybe, I will find some interesting information that I can share with them to give them some other perspective outside of academia. And after talking and talking, at some point, I just realized well, this is not that unachievable that I thought. So why not just trying for Robin? The foundation has all their budget already you know, put in those research, they cannot put, you know, more elsewhere. So I just thought, you know, let's just try this. One day I came back home and I told my husband, like, why didn't we do that for him? He just didn't understand where that was coming from, but that was funny.
So let's take a step back and talk, what is a rare disease? How do you work on a potential treatment? Like, what's the definition and how do you approach working on this? And we had a comment in the chat that the particular diseases, very few people are sick with them, but in total, it's a large number of people. So maybe you can elaborate on that.
Exactly. I really like this chart from the Nord, which is, yeah, there are 7,000 rare diseases around the world, which is, well, 25-30 million Americans, but it's worth 300 million worldwide, estimated, of course. It's defined, at least in the US, the definition is different in Europe, but in the US, it's a condition affecting fewer than 200,000 people. In Europe, it's fewer than 1 out of 2,000 people. But so definitely, you know, a lot of them are pediatric diseases. And why are they so difficult to cure? Because they are so rare. I mean, that's definitely the number one. I mean, which pharmaceutical company will be interested in marketing something that is for 100 patients, right? None. So what is happening?
I don't think that's something you do for money.
Well, yeah, exactly. But what is happening today also is that the rare disease community, instead of staying alone, each disease, they try to gather, you know, to find a common place to be a larger community, you know, to come to pharmaceutical companies with a larger community. So when you think rare disease, you think of those 3 things I just listed, you know, gene therapy, which is in the strict sense, because oligonucleotides can be considered gene therapy, but in the strict sense, it's those gene replacement therapies. So pretty much you introduce a new gene within the DNA, which is either a functioning when you have a non-functioning one, or you try to block one that is not desired by introducing a new one. So this is a very long process. This is really de novo drug development. You know, it takes time, and I'm not saying it couldn't be done for Cat6, but certainly not right now because the community just doesn't have the funds to do that. Oligonucleotides are obviously a way also, as I explained, to act on these. And the reason why I was saying this is not for Robin for now is that Robin doesn't have anything that needs to be silenced. His mutation actually results in one of his copies of Cat6b to be inactive. So his problem is that he just doesn't have enough of it. So we would more look into boosting the gene expression of the one that is working rather than trying, you know, to silence anything, because we don't have anything to silence. There's no splicing thing also, but oligonucleotides are being explored also for some additional things. So maybe later, you know, it will come, but not for now. And so there is the repurposed drug screening, which I will describe also a bit more after.
How can treatment be?
I see we are running late, that's why.
Yeah, so we are respectful of your time. We would love you to stay with us till the end because we have a couple of things to share still. So please do if you can. If no, go ahead and watch the finish as a recording. We would very much appreciate that. So with that being said, how do you personalize medicine for something that rare?
Well, so personalized medicine is not a new thing, you know. It's been ongoing for like quite some time now, especially in the cancer treatment area, because it really comes from the fact that it has been long observed that you can, you know, people with a treatment, people will react differently with the same treatment. You know, even for simple treatments like taking acetaminophen, you know, it will take 2 for you, it will take 1 for me for the same thing, right? Because we are all different. And this is pretty much related to different gene and protein expression profiles, you know, in individuals throughout the population. And so the concept of classifying people in different groups based, for example, on their gene profile, it can be also biomarkers in the blood, in the urine, but let's take gene profile because that's what it is of interest for us. Well, this concept has emerged as a way to adapt treatments for a maximum efficacy of those treatments. And so this principle can certainly be further applied to rare diseases, you know, especially, you know, Cat6b, as I was saying, it's not like some recessive rare diseases where, you know, every individuals, you know, the exact genetic mutation that they have, you know, they all have the same one and that needs to be corrected. Well, for Cat6b, that's not the case. So obviously it brings another challenge, right? And that's why, you know, gene therapy, oligonucleotides, they would take too much time. And nowadays there is also drug repurposing that has emerged also as a very effective way to find new therapies, you know. And the principle, again, it's very simple and it's not new, you know. Drug repurposing has been based on side effects observed, you know, for many years. This is why Viagra came into the picture, right? It was supposed to be for cardiovascular diseases, but at some point they realized that there was some side effects that they did not expect that seemed to be promising. So they marketed it as for another purpose, you know. And as I was saying, you know, you can see here on the left what, you know, drug development from scratch takes, you know, it takes 10 to 15 years, it takes billions. The success rate, obviously, you know, from start to which drug makes it actually to clinical trial is very low. Whereas you have all these drugs that are all already on the market that are intended for one indication, but you never know, you know, they could definitely be useful for other things. And this would just shorten, you know, it's already on the market, the safety has been assessed. There are so many steps that can be skipped, you know, if one of these could be used. And so that's what definitely we're aiming to. And now we also have, you know, tools to really screen a high number of drugs at the same time. So what really we are looking for Robin is really simply take a skin biopsy on him so that we have his cells with his mutation, and those cells will be cultured and characterized, you know, to see exactly the kind of defects that they have, to then, you know, try some drug repurposing. And ideally, you know, at some point like this chart shows, you want to be able to transdifferentiate the cells into neurons, because working on neurons is the best, you know, especially for a disease like his. But that's another step, even much more expensive. So let's go for the base for now. And this is what we want to achieve with high throughput screening. Basically, you have libraries of drugs that are available, and you just put them all on, you know, a small place containing the cells, and you just, simply said, you just see if there's a hit or there's no hits, right? But it takes, you know, all these phases I am showing up there. First phase is the development of an assay. You want to make sure that the assay you will use to actually tell you, yes, this drug is a hit, no, this drug is not a hit. You want to make sure it is accurate, it is reproducible. And with Cat6b, we don't know where we are going. You know, we need to try different things to determine which assay will be the best. And I just put the diagram under that to show that there are different ways to read out the assay. You know, it can simply be just solutions and it just lights up, like on the left. High content imaging will really take pictures of the cells, and you just see between the left and the right panel of the cells that there's a difference. You know, there's more on the right within the cells than there is on the left. So those are the kind of readouts that you get from those screenings. And the assay development itself pretty much is already a couple $100,000, you know, just by itself, because it's the most important part. And then once you validated that, you start a pilot screen, which is just a small number of molecules to, again, make sure your assay responds the way you expect it to respond. And then you can finally do your primary screen, which is the whole library of drugs. And then there is another phase after that, because once you have hits, right, you know, some drugs, you know, you had a positive result. Well, you need to determine which concentration needs to be used. And again, some of these drugs will fail that test because the concentrations needed will be too toxic. So we won't be able, they may be hits, but we won't be able to use them. So pretty much it's from what I've heard, been told actually by a parent, another parent going through that, is that you get out of your screening, you get 5% of the drugs that are hits. Then out of the 5%, it's another 5% that will actually have a real efficacy for what you're looking for. And then it's another 5% of those that will be actually usable at a decent concentration, right? So that's really our project. And oops, sorry. I just wanted to, you know, finish this with just a simple fact that, you know, Robin still has hospital appointments. As you can see here, he was diagnosed this year with epilepsy. So he needed to be treated for that. But I think it's really important to, in all of this, keep a good balance, you know, keep a happy family, obviously, but keep a good balance of things, because I literally missed the first year of Robin. I was so into taking him, you know, invested in the therapies that I just simply forgot to be a mom, you know, and I realize it now. I don't regret it because that's how it was, but, and it's difficult. I mean, those times are certainly difficult, but this balance I am talking about is really important because it's a marathon, you know, it's not a sprint. You're in for a long run. So that's what's important, I think, ultimately. And I'm very happy to have shared this today.
Definitely. Thank you so much. Because we're out of time, I'm just gonna have this big call to action to you. I'm pasting the link to Typhaine's fundraiser now in the chat. So I donated to this fundraiser, but as I said, there's only so much one person can donate, but many people can donate a lot more. And Typhaine told me about the fundraiser that Michael's parents, the fourth kid you saw on the slides, are having, and they are looking for millions, and they already have millions because many people donated little small amounts. So I have now this routine I developed. When I take my kids to the doctors, I go and get myself a Starbucks and the Starbucks coffee, and then I get a cake pop for my kid. Like, what the heck is even cake pop? He loves it. Anyway, I decided that if I can afford this overpriced treat for myself and for my kid, then I can also afford matching this every time I do it for this fundraiser. So that's what I did yesterday. I sent Typhaine a little picture of my Starbucks receipt, and she's like, huh? What are you doing? So this is what I'm doing, and please do it as well. If most of us can afford the coffee, that probably doesn't taste different than the coffee that we make at home, but we still pay like $5 or $10 for this, that means we can afford to help as well. So go ahead and donate. And thank you so much for joining, and thank you so much for staying. A lot of people stayed over time. I know we are all late for meetings, so I will be sending you the recording. Thank you, Typhaine, for joining.
Thank you everyone.